My name is Sarah McMullen, and this is my Exam 4-1 video for Genetics 311 on point mutations. This is the original non-mutated strand. Given the original code, make sure to write out the transcribed strand before translating into mRNA strand. Using the codons from the mRNA, you can determine the amino acid sequence. A point mutation occurs when one of the bases is substituted for another base in the coding DNA strand. The first mutation we will discuss is a missense point mutation. The base pair that changes in the original coding DNA strand causes a different amino acid to be sequenced from the mRNA strand. The changed base, as well as the corresponding changes it causes, are shown in red. This base change is a transversion since the base A, a purine, is replaced with the base T, a pyridamine. To easily demonstrate what happens, we are going to correlate the amino acid sequence to flower stems. In the pictures here, you can see the original flower stem compared to the missense flower strand, which is curled due to having a different amino acid sequence. A nonsense point mutation occurs when the change in base in the DNA coding strand causes one of the amino acids in the sequence to become a stop codon, stopping the sequence prematurely. In this example, the base C, which is a pyrimidine, is changed to a base A, a purine, in a transversion mutation. The changes are shown in red. Since the amino acid sequence is cut short, the flower stem for a nonsense flower is shorter when compared to the original unmutated flower stem. A silent point mutation is when a base changes in the coding strand, but still codes for the same amino acid in the amino acid sequence. The differences caused by the mutation are shown in red, but as you can see, the amino acid sequence doesn't change from the unmutated sequence. This base change is an example of a transition change, since the base T, a pyrimidine, is being changed into base C, which is also a pyrimidine. Though these flowers have a slightly different coding strand, they code for the same amino acid sequence and therefore have the same flower stem length. The last type is a read-through point mutation. This mutation occurs in the three bases that will code for a stop codon. The change in base causes a mutation that codes for an amino acid instead of a stop codon, continuing the amino acid sequence further than it originally went. This base change is an example of a transition change since the base T, a pyrimidine, is being changed into base C, which is also a pyrimidine. As you can see with the flower stems here, the read-through flower stem is much longer than the original flower stem since its amino acid sequence continued to code due to the mutation. I hope this helps you to better understand point mutations. If you need further assistance, reference Dr. Halweg's PowerPoint slides or Genetics: A Conceptual Approach. Thanks for watching.